All right, I guess we're rolling here. So, hello everybody. My name is Elliot Kimmel, and this is my exam review for grade 12 biology. This is just going to be a very casual video tutorial. I'm going to cover a lot of stuff related to the grade 12 biology course, reviewing for the exam. So it's going to require some prior knowledge, and hopefully I'm going to go kind of quick through a good portion of this. All right. So there's a couple of things that I want to touch on here, and one of the first things I want to talk about is cellular respiration, in particular glycolysis. So I've got an image here that comes from the Nelson biology textbook, and I hope that it's not too fuzzy for you to see. So the idea is that glucose up here is the main fuel for cellular respiration. Of course, you can use others. You can use proteins, you can use lipids, and they all feed in at various points in glycolysis or in the Krebs cycle or whatever. But we're going to start with the glucose, and this is going to occur in the cytoplasm. It does not require oxygen, so it's anaerobic respiration. And when the glucose is in the cytoplasm, it's going to encounter some ATP. So an ATP molecule here bumps into the glucose and phosphorylates it. So this is the addition of a phosphate group from the ATP onto glucose, and it's going to go on the 6th carbon. So here is the 6th carbon, and there's the phosphate group. And so that's going to leave us with a molecule of glucose 6-phosphate. And of course, we've used one ATP, so we now have ADP. Glucose 6-phosphate is going to be converted into fructose 6-phosphate. That's an isomerization reaction. And then fructose 6-phosphate is also phosphorylated by ATP to produce fructose 1,6-bisphosphate. So now we have two phosphate groups. There's one here and there's one here. Okay, so this is carbon 1, this is carbon 6, so fructose 1,6-bisphosphate. That molecule will then be cut by an enzyme or a few enzymes. And because those sides aren't exactly the same, although it looks symmetric here, there's actually functional groups coming off those carbons, we're going to get a molecule of DHAP, dihydroxyacetone phosphate, and we're going to get a molecule of G3P, glyceraldehyde 3-phosphate. But the cycle uses G3P, not DHAP, so we have one here and we have one of these. Well, this one is going to get converted into another glyceraldehyde 3-phosphate, so that we'll end up with two of them. Each of them will then continue down in the biochemical pathway. All right, so let's just follow one of these G3Ps. G3P encounters a phosphate group that's floating around in the cytoplasm. This is not attached to an organic molecule, so this is called inorganic phosphate, which comes on, and this creates 1,3-bisphosphoglycerate when the phosphate group comes on, and NAD+, this is an oxidized form of an electron carrier, nicotinamide adenine dinucleotide, NAD+. When it gains electrons and hydrogen from G3P, it then becomes reduced to NADH, and we'll see the use of NADH later in the electron transport system, and that leaves us with our product of 1,3-bisphosphoglycerate. Now, this molecule finally phosphorylates ADP, producing our first ATP. Now, there are two of them made because remember we had two G3Ps, each going through the cycle, and we now have produced two ATP. This is called a substrate level phosphorylation because the phosphate group going on to ADP comes from a molecule in the biochemical pathway. In other words, this molecule here that gives the phosphate group is a substrate for the enzyme that would cleave the phosphate group here and put it onto ADP to produce ATP. So this is called substrate level phosphorylation of ADP, right? Because we're making ATP. And that leaves us with 3-PG or 3-phosphoglycerate. We only have one phosphate group left, and we've got a 3-carbon molecule here. 3-PG is converted into 2-PG, another isomerization reaction. 2-PG releases water, releasing PEP in the end, phosphoenol pyruvate, and there is one more substrate level phosphorylation onto ADP to produce ATP on both sides, and that leaves us with our final product of pyruvate. Pyruvate is a 3-carbon molecule, and because there are two of them produced, these two 3-carbon molecules add up to the original 6 carbons that were in glucose. So the net result is the production of 2 ATP, 2 pyruvates, and 2 NADHs, one here and one here. All right, so that's the process of glycolysis. Let's go on and have a look at another picture. So this is an image of glycolysis as well, and I just wanted to show you this one because it has the structures and it also has the names of the enzymes here: hexokinase, phosphoglucose isomerase, etc. All right, so once again, we can see glucose converted to glucose 6-phosphate as ATP phosphorylates the glucose. Glucose 6-phosphate to fructose 6-phosphate, the isomerization, and notice the enzyme isomerase. And then another phosphorylation to producing fructose 1,6-bisphosphate, the production of DHAP, and complicated enzymes are G3P here, glyceraldehyde 3-phosphate. And here we can see another way that the redox reaction can occur, NAD plus going to NADH. Note the hydrogen here. So, I mean, some people write this as, I've seen this as NADH2 before as well, although I don't really like it that way. Anyhow, we have that reduction reaction there. We have the phosphate group added on, and we just continue down the pathway. But of course, we're producing the ATP there. We're producing the ATP there. We're getting our pyruvate, and we're getting our NADH right there. And that's the way it goes. All right, so there's glycolysis for you. And let's move on. Now, the next step is to take the pyruvate here in the cytoplasm still. So we're up here in the cytoplasm, and that will then enter the mitochondria. And this is where the majority of ATP is going to be produced. So this is why the mitochondrion is considered the powerhouse of the cell, because it's going to create a lot of ATP for us, aside from the, the 2 ATP that was produced by glycolysis in the cytoplasm. So the pyruvate will enter the mitochondrion and undergo pyruvate oxidation. So we are going to oxidize the pyruvate and convert it into the product called acetyl-CoA. And acetyl-CoA will be the starting molecule for the Krebs cycle. So collectively, this and this are the 2 main reactions that are called matrix reactions. So in pyruvate oxidation, carbon dioxide is removed. That's a decarboxylation. That's going to take this from a 3-carbon molecule down to a 2-carbon molecule, forgetting about the oxygen for the time being. This is some of the CO2 that you're exhaling right now and that I'm exhaling right now. All right, the loss of the CO2. And NAD+, the oxidized form of the electron carrier, will be converted to NADH. And notice again that this textbook writes the NADH plus H plus form, but we're really not too concerned about this, although this can be important for hydrogen ion concentration in the mitochondrion. But for our purposes, we care about the NADH. So we've oxidized the pyruvate by reducing the NAD plus to NADH. This will be used in the ETS coming up. And now we have our 2-carbon acetyl-CoA molecule. Now we go into the Krebs cycle. Acetyl-CoA will bind with the 4-carbon acceptor molecule, oxaloacetate, so the 2 are going to collide here, and that's going to produce a 6-carbon molecule of citrate. Now, because this is the first organic molecule in the pathway, this is also called the citric acid cycle. Citrate and citric acid, same thing. Citrate will then be converted to isocitrate, an isomer. Isocitrate is then converted to alpha-ketoglutarate as a carbon dioxide is removed. So there's another CO2 that you exhale. And there's another redox reaction, NAD plus floating in the matrix, right? This is the liquid jelly-like interior of the mitochondrion. NAD plus will pick up electrons and hydrogen from isocitrate, converting it into alpha-ketoglutarate. And in the process, it is becoming reduced to NADH. Again, we're going to use this in the ETS. Alpha-ketoglutarate undergoes a similar reaction as CO2 is released that you exhale, another production of NADH, and we now have succinyl-CoA. Succinyl-CoA is converted to succinate in this complicated reaction where the CoA group is removed, so it's succinyl, we get rid of that CoA there, now we have succinate, and we have the entry of another high-energy molecule that does a sort of handoff, producing our ATP. And we're really just going to focus on the production of ATP down there and not worry too much about these other high energy molecules. So we've produced 1 ATP per acetyl-CoA coming into the cycle, and that's all. But of course, remember that because there are 2 pyruvates from glycolysis, we're going to get 2 acetyl-CoAs, and eventually we're going to get 2 ATPs, all right, from the original glucose that went through glycolysis and then through this. So succinate is converted to fumarate as a new electron carrier, FAD, flavin adenine dinucleotide, picks up electrons and hydrogen, and is reduced to FADH2. This also enters the ETS and will generate ATP for us. Now we have fumarate, and fumarate gains some water and is converted to malate. And note that we have a 4-carbon molecule here and here and here because we lost a carbon there and we lost a carbon here. So we started with a 6-carbon molecule up here. We're down to a 5-carbon there. We're down to a 4-carbon after we lose that CO2. We're at malate. Malate then reduces NAD+, producing NADH again so that we can use that in the ETS, and we're back to oxaloacetate. So this cycle, which goes round and round, churning off this NADH, a little bit of ATP and CO2, really starts and requires the acetyl-CoA coming from your food, from the original glucose, and then it creates these products for us. All right, so from the original glucose, making the 2 pyruvates and then 2 acetyl-CoAs, we get a number of carbon dioxides. We've got 1 there, we've got another one there, we've got another one there. So per glucose, we're going to have 6 CO2s because there's 3. 1, 2, 3. We get 6 CO2s. We are going to get 1, 2, 3, 4 NADHs, again, per glucose, right? And we're going to get 2 FADH2s, and we're going to get 2 ATP. All right, so we haven't created a lot of ATP yet, although we're in the mitochondria, and that's supposed to be important. We're about to do that now. All right, we have now basically used up all of the carbons and gotten a lot of energy out of our glucose, and it's time now to move to the electron transport system. So this is the diagram from the Nelson textbook. Let's choose an interesting color. Let's go for some pink, weird pink. All right, where do we pick up here? Pyruvate from glycolysis in the cytoplasm comes in. Okay, we got our acetyl-CoA. This is showing the inside of the mitochondria, the mitochondrial matrix. We went through Krebs cycle. We produced some NADH and some FADH2. We're about to use that stuff. We released some CO2. We made some ATP. And by the way, this was substrate level phosphorylation because the phosphate group to produce that ATP came from a substrate, a molecule within the Krebs cycle pathway. So it was substrate level phosphorylation, as opposed to the oxidative phosphorylation that we're about to see here. So these electron carriers here and the NADH produced in the pyruvate oxidation, which is in the matrix, are going to deposit their electrons into the electron transport system or electron transport chain, which is along the cristae of the mitochondria, along the inner membrane. You see here it says inner mitochondrial membrane. So that's where these components, various proteins and other molecules, globular molecules, are located. So let's have a look at this NADH. It floats over to the first ETS component or carrier, which is called NADH dehydrogenase. So it takes NADH and it dehydrogenates it. It takes the hydrogen and the electrons off. The hydrogens are going to be pumped out of the matrix into the intermembrane space or the intermembrane compartment. And the electrons are going to be passed along this red arrow down the ETS. All right, so it takes the electrons, keeps the electrons in the chain, pumps out the hydrogen there. And we can see that 1 hydrogen is pumped out. Now, those electrons will then flow from carrier to carrier, Q, cytochrome BC1 complex C, and cytochrome oxidase. They will flow down the ETS or be handed off from carrier to carrier in a series of oxidation and reduction reactions. In other words, when NADH gives electrons to NADH dehydrogenase, that reduces the NADH dehydrogenase, but it oxidizes the NADH. And this, of course, goes back to NAD plus and can be reused in the pyruvate oxidation step. Or if this is the NADH dropping off electrons, this goes back to NAD plus and can be used again in Krebs cycle. So the electrons travel from carrier to carrier, and as they do that, they release their energy. That energy is then used at 3 spots to pump hydrogen ions that are inside the matrix, either from the NADH or just floating around in here like this one looks like and this one looks like, and it pumps them into the intermembrane space, and it does it at 3 sites. That builds up the concentration of hydrogen ions. All right, this is creating a proton motive force. A high concentration of hydrogen. Of course, it's going to be decreasing the pH, making it more acidic. Anyhow, those electrons get to the final ETS component called cytochrome oxidase, having given up their energy to pump the hydrogens, and they then combine with some of the hydrogen that is in the matrix or re-enters the matrix through here. All right, this hydrogen could come in here, or it could be this hydrogen, and also combines with oxygen. This is the O2 that you breathe. So you breathe in oxygen through the respiratory system, transported on hemoglobin of red blood cells to the cells that are undergoing cellular respiration, in through the cell membrane, into the mitochondrion, and then finally the oxygen is available. Now, of course, there's going to be enzymes like this one here and various other things that are going to make this reaction happen so that you can unite those electrons, the electrons that are here, with hydrogen and oxygen and produce water. And water can then just diffuse by osmosis out of the mitochondrion, and the electrons have done their job. They have built up the hydrogen ion concentration. Now, the oxidative phosphorylation part, the part that involves ATP synthase, has the hydrogen ions that are high in concentration, which are charged and cannot just re-enter the matrix through the phospholipid bilayer here. All right, charged things cannot do that. They need to travel by active transport, facilitated diffusion, something like that. Well, this is facilitated diffusion in that the hydrogen ions are in higher concentration here than here because they were built up. So they will then travel through this pore called an ATP synthase with the enzyme here, ATP synthase, or this whole part could be considered the enzyme. And as the hydrogens flow from high concentration to low concentration, that's facilitated diffusion, they stimulate the enzyme to convert ADP and PI floating in the matrix and convert that into ATP. Because imagine this stuff, ADP is like a car with no gas. All right. And PI is the gas. All right. But they're separate. So the car doesn't run. But when the hydrogen goes through and activates the enzyme here, the gas goes into the car and now it's together and the car runs and it's now called ATP. It's a bad analogy, but you know what I mean. All right. So every time one NADH drops off its electrons, 3 hydrogens are pumped from the matrix to the intermembrane space. They diffuse back in, and each one produces an ATP, and therefore each NADH can produce 3 ATP. All right, because 3 hydrogen ions were pumped to the intermembrane space, so 3 ATP are produced. So we can now already see that. I mean, that's more than glycolysis itself, right? 2 versus 3. So the mitochondrion is making the majority of the ATP. Now, FADH2. Let me clear this up and switch to a different color. FADH2 over here is going to deposit its electrons at the second ETS carrier or component. It misses the first one, and therefore it misses the first site of hydrogen ion pumping. It drops off electrons at Q. That, of course, regenerates FAD, which goes back into Krebs cycle. All right, and the electrons then travel down the chain and only lead to the pumping of 2 hydrogens into the intermembrane space. They re-enter the matrix, and that leads to the production of 2 ATP instead of the 3. But still, from that one, acetyl-CoA coming in, we've created a lot of ATP. All right, so that is the electron transport system. That is oxidative phosphorylation. Sometimes this is called the chemiosmotic model for ATP production. Now, let's see if I've got another image for that as well. Yeah, I happen to have one more that I wanted to show you, just so you see it in a few different ways. And there's going to be maybe some different terminology. So here we have NADH. Don't worry about this other stuff right there, all right? Carrying electrons from the food. And the NADH is dropping its electrons here at step number 1, the first carrier, which would be NADH dehydrogenase. Hydrogen ions are being pumped into the intramembrane space, regenerating the NAD plus to be used again. Those electrons are traveling down, releasing energy, causing the pumping of hydrogen ions. That's why they're drawing the plus signs there, because positive charge is building up outside of the membrane here. And then those hydrogen ions, 3 of them, will then one at a time come back through the ATP synthase and lead to the production of ATP. At the end, electrons go on to hydrogen and oxygen, producing the water. And here you can also see the FADH2 converted to FAD for reuse, again, as it drops off electrons to Q, causing two hydrogen ions to be pumped out and the production of 2 ATP. All right, so I hope that helps with that concept. Let's move on. All right, we're completely switching gears now. That was a little bit of a review of sort of cellular biochemistry, metabolism. And there's lots more, of course, in the course, and you need to go through your notes and know it as well as possible. And it's a challenge. We're moving into the homeostasis unit, which was divided into 2 parts, the nervous system and the endocrine system. The functional unit of the nervous system, of course, is the neuron. So here we go. And I think I'm in PowerPoint, so yeah, don't really want that there. Let's see if I can get rid of that. Good, get rid of that. All right, am I writing again? Good. Okay. So we've got the neuron. I think I just wrote that, didn't I? Let's write it again. All right, the neuron is the functional unit of the nervous system. And of course, a neuron here, this is a cylindrical neuron, starts off at rest. So it's at resting membrane potential, or RMP, which is roughly minus 70 millivolts. And you can see that over here on the graph. And that is going to be due to the differential concentration of ions, the permeability of the membrane, and the sodium-potassium pump. And I hopefully have covered that pretty well in class. So I just wanted to have a quick look at the action potential. There are different ways of drawing this. I mean, they're very similar, but sometimes they leave off the graded potential at the beginning, although this one has a little bit of a bump there. So let's go through the graph. Membrane potential is measured in millivolts versus time in milliseconds, and this curve is called an action potential. And when a neuron is stimulated, so for example, if you stimulate it there, or in other words, if we stimulated a neuron by poking it or applying some stimulus like light or heat or sound or some kind of deformation, this is going to cause a graded potential. And a graded potential is when a small number of sodium ions, usually, leak into the neuron through leak channels, bringing with them their positive charge. And now normally the inside of the membrane is negative and the outside is positive, and that's set up in resting membrane potential, and that's why we see this minus 70 millivolts. When a little bit of that sodium leaks in, we start to get a depolarization and the curve starts going up. If enough of those sodiums leak in to hit a particular point, which is called threshold or threshold potential, the action potential is fired. So although this is called the action potential, the prelude to it, I mean, this region here is essentially the graded potential. So once you reach threshold potential, this is where the voltage will rise very significantly. And it's going to go from around minus 55 and overshoot it to about plus 30. So we're negative inside. That's what this is showing here, right? We're starting off negative, minus 70, and then around minus 55, so it's gotten a little bit more positive, but it's still negative. It's going to shoot up, and you're actually going to get a bunch of positivity on the inside. And that's because voltage-gated sodium channels will open, so we have sodium again, and we'll get a massive influx, a lot of sodium coming in, a large influx of sodium coming in, causing the voltage to rise. All right? So this was the opening of the voltage-gated sodium channels. The voltage-gated sodium channels will then spontaneously close a fraction of a second later, like you're looking at, 1 millisecond or 1.5 milliseconds. They will close and the curve will plateau. No more positive charges coming in. In addition to that, voltage-gated potassium channels will open, and there will be a large K plus efflux. And that's because potassium is in high concentration on the inside. That's pretty messy. I hope you know what I mean. So we're going to close the voltage-gated sodium channels, open the voltage-gated potassium channels. As positive charge exits the neuron, it starts to become less positive inside, so we're going down, and essentially more negative. So we're going to go down here, and then we're going to come back to rest as the sodium-potassium pump reorganizes the correct ion distribution. All right, so this is called the action potential, and it occurs really in a very small region of the neuron. You know, a small area. It's not spread out over a large area. I mean, if you stimulate right there, this is where all that happens, but I drew it over a larger area. And so it's essentially a brief reversal of the charge. You go from minus on the inside to positive, back to minus. And that's it. Minus, up, back down to minus. That's an electrical change, and that is the signal that is going to be propagated down the neuron, all right? Not the graded potential, which triggers the action potential, but the action potential itself. And it doesn't matter how much of a stimulus you give to the neuron, the same size action potential is going to occur, because once you reach threshold, those voltage-gated sodium channels open, and they will spontaneously close like a door that you push open and it just comes back on itself. It's just going to close. So if you stimulate harder, it's not going to go up higher for a particular neuron. It's going to go the same magnitude, and it's going to take the same amount of time to get back to rest. It's the same duration, around 3 milliseconds or so. All right, so that's how the action potential is formed. And then now let's look at the conduction of the action potential. All right, time out. Sorry about that. So what we want to do here is we want to look at two different types of neurons. We want to look at neurons that don't have myelin sheath on them and neurons that do, because the myelin sheath is this structure here. It's created by Schwann cells that are somewhat flattened, and they wrap around the axon, and they provide an insulating cover. And therefore, the axon itself is only exposed at various regions called nodes, or nodes of Ranvier. So we're going to come back to that in a second. Let's look, first of all, at the or non-myelinated neuron. So what we have here is negative on the inside, positive on the outside. That is our neuron at rest. All right? And when we stimulate the neuron, as you can see by these arrows here, positive charge enters the neuron through those voltage-gated sodium channels, and that reverses the polarity of the membrane. It's positive on the inside and now negative on the outside, relatively speaking. The rest of the neuron, though, is still at rest, at resting potential. This entry of positive charge will then diffuse down to the next region, let's say right adjacent to it, and if possible, depolarize this region to threshold. When that happens, positive charge will rush in, and this will flip to that, and now the action potential is moved from there to there. This positive charge will then move to the next region, which is at rest, depolarizing it to threshold, positive inside, negative outside, and now an action potential occurs here. Of course, a fraction of a second later, the positive charge exits. Voltage-gated potassium channels, right? And same thing will happen here, and it'll go back to rest. But this is how the action potential is propagated or transmitted on a non- or unmyelinated axon. It occurs at every spot along the neuron. If you imagine a meter stick, right, and you've got millimeter markings, just imagine it occurring at 1 millimeter, 2 millimeters, 3, and it just keeps going like a burning fuse. It just happens again and again and again and again. And it's very, very quick, but it's not as quick as this next method we're about to look at. All right, so the action potential started up here, and it was propagated down or transmitted down to the end of the neuron, and it gets down to there, and maybe from there it's got to jump across a synapse, or maybe it arrives at its destination. Now, if we compare that to the myelinated axon, the pumps and channels are localized to the nodes. This saves in energy and resources to build them there. There's no sense having those channels of voltage-gated sodium and potassium channels and sodium-potassium pumps, no sense having them under the myelin sheath because they're useless there. So this is where the pumps and channels are, and this is where the action potential occurs. And this diagram shows a node that is undergoing an action potential. All right, because it's positive on the inside. Normally it's negative on the inside, right? Like over here, it's positive. So sodium came in, it reversed the polarity, and now that is going to affect the next node. Now notice that this node here is at rest right now. It's still around minus 70 millivolts on the inside, but when the sodium that entered diffuses down the axon, it can't escape through the myelin sheath, because myelin is a lipid chemical, and sodium is positive. And just like hydrogen couldn't go through the inner membrane of the mitochondrion, sodium can't go through the myelin sheath. So it's prevented from escaping so that it stays within the axon and makes it to the next node, depolarizes this node to threshold, and an action potential fires here. When that occurs, sodium's going to come in, flipping the charge here. That sodium will then diffuse down to the next node. So what we get is an action potential jumping from node to node, and this is a faster method of propagation or transmission than on a non-myelinated neuron. And basically you're seeing the same thing here. We see the influx of sodium here, the sodium diffusing to the next node, causing an action potential, then the sodium diffusing to the next node where it will occur. So far we're still at rest right there. All right. Now, by the way, one thing to notice is that when sodium influx occurs right here, all right, when the sodium comes in, the sodium doesn't have to diffuse to the right. The sodium can diffuse in any direction. Of course, it can't get out through the myelin, so that's useless. But if it happened to diffuse to the left, let's say, which just fired an action potential a fraction of a second earlier, you might think that the action potential will go in this direction and an action potential will go in this direction, which would be weird, right? It's like, you stimulate your hand and it goes up and then it goes back down your hand. That doesn't happen because this region that just fired an action potential is in its refractory period. And at that point, the voltage-gated sodium channels are actually locked shut so that even if you got a suprathreshold stimulus with lots of sodium, all right, the voltage-gated sodium channels won't work, and you therefore will not get an action potential going back in that direction. By the time the voltage-gated sodium channels are ready for a new stimulus so that action potentials can go down the neuron again, by that time, the voltage-gated sodium channels are primed and ready to open, and by that time, the action potential here is far down the neuron, and there is not enough sodium to ever make it all the way back. All right, so there we go, propagation. Where are we going to now? All right, we are switching gears completely. We are going to start talking about genetics. And in genetics, one of the first concepts that we cover is replication of DNA. DNA needs to be replicated for mitosis or meiosis so that chromosomes can go into daughter cells. And we've got 46 chromosomes here to be replicated. I don't know if I'm writing actually right where my picture is going to be, so 46 chromosomes that need to be replicated, and the way this can occur is, if you have your typical DNA molecule here, let's say, there's your DNA. You might think of unzipping right down at the bottom, all right? But it doesn't have to happen that way. You can have all your bases here, and replication can start here, and replication can start here as well, with a number of different enzymes. And that's what this image is trying to show, the fact that the enzymes here involved here, helicase for breaking the hydrogen bonds, can start somewhere within the actual DNA strand. And if that occurs, it separates the strands, and then the enzyme can move to the left here, or it can move to the right, or actually, and it moves in both directions. And this is called a replication bubble right here. All right, so these are replication bubbles as the enzymes move in both directions, creating this bubble-like structure. And of course, they're going to meet here, and then the whole DNA is unzipped. Now, this diagram is just a little bit more typical. What would have happened is this top strand up here and this bottom strand in pink or purple would have been joined together at one point. The enzymes came in here, gyrase to untwisted, relieve some tension, helicase, and here we can see DNA helicase right there to unzip it, to move to break the hydrogen bonds and separate the complementary bases. And then single-stranded binding proteins attached to prevent these bases from reannealing, reattaching by complementary base pairing. and then we now have the free DNA strands so that we can start replicating. In this case, DNA polymerase, which we see right here, attached down here, and it moved in this direction, and this is called the leading strand, and it will produce a DNA strand in continuous replication. This is continuous DNA replication because the DNA polymerase attached, it's building a new strand in the 5' to 3' direction with respect to that new strand, right? That's important. It's 5' and it's 3', and these are the complementary bases that were floating around in the nucleus. And so when DNA polymerase saw an adenine, it grabbed the thymine and clicked it on. When it saw cytosine, it grabbed the guanine and clicked it on. So it moves in this direction, putting on the nucleotides, complementary base pairing, creating what's called the leading strand. On the other side, DNA replication cannot also move in this direction, all right? It has to build in the 5' to 3' direction. So it attaches, let's say it started here, and it built a strand in the 5' to 3' direction. And then helicase unzipped the DNA a little further, maybe up to here. And so a new DNA polymerase attached, going in this direction, producing another piece. Excuse me. These are called Okazaki fragments because DNA replication is occurring in a discontinuous manner, and they are not attached initially to each other, and they will be attached by the enzyme DNA ligase. So this is discontinuous replication, and this is called the lagging strand. On with the show. So this image shows an overview of the process. Once again, we've cut open the DNA here. We have DNA polymerase synthesizing a new strand in the 5' to 3' direction continuously. That would be the leading strand. Here we see that DNA polymerase. And here we can see a couple of different DNA polymerases attached on the other side, synthesizing in the other direction, making Okazaki fragments, which will then be joined by DNA ligase. All right, this will eventually get to the end, and we will have a DNA molecule here that is identical to the DNA molecule here, and that is how we replicate the DNA or replicate the chromosomes. And by the way, this is semi-conservative replication because the original parental strand, this purple one here and this purple one, is now a part of the daughter strand, which has an old parental strand and a new strand newly synthesized. So it's semi-conservative because half of it existed before to create the final daughter DNA. Okay, you got a parental strand and you got a daughter strand. So it's semi-conservative replication. So there's DNA replication in a nutshell for you. And that brings us to using the genetic code to create proteins, polypeptides, all those hormones and enzymes that you learn and stuff like that. So, of course, DNA has the genetic code. And here we can see a little piece of DNA with a bunch of letters. These are the nitrogenous bases. The genetic code is in the order of the bases, and it's read 3 bases at a time. So here's 3 bases, and that means something, and that 3 bases means something, and that 3 bases means something as well. So the genetic code is in the DNA. The DNA, of course, is in the nucleus. It's far too precious to come out of the nucleus of that cell to create the proteins, polypeptides, whatever. So a copy of that DNA code is produced, and this is called transcription. The process of transcription converts the code of DNA into a complementary sequence of bases in a molecule called mRNA or messenger RNA. This has RNA nucleotides, so it's got ribose sugar, it's got uracil instead of thymine, and it's got some other structural differences as well. So the code of DNA is copied into mRNA, and that is called transcription. The code that is in mRNA now needs to now be translated. So the process of translation converts the 3 base sequences in mRNA, the codons, into specific amino acids. Those amino acids are linked together by peptide bonds, and this forms the final polypeptide, and that's called translation. All right, so DNA to transcription, then the translation of that mRNA, and then the production of our final protein, which might be polypeptides initially that have to be linked together. This diagram shows DNA unzipped because let's say this section of the DNA right here, between here, is our gene. And one gene will code for one protein or one polypeptide according to the central dogma. So the DNA is a long stretch, of course, and there's lots of genes on that, but this will unzip at that particular location, and we will say, okay, we need to make a copy of this area because this is our gene, and it's going to make our polypeptide product, so let's make a copy of this in transcription. That's going to require the enzyme RNA polymerase. That's the enzyme that recognizes the DNA base, let's say cytosine, and grabs a guanine ribonucleotide. So it's going to have the guanine part, whatever, it's going to have phosphate, it's going to have ribose sugar. And, it's just going to move down, and it's going to make a complementary strand, but RNA instead of DNA. And then this RNA, we are going to assume for now that it's messenger RNA. Of course, it could be transfer RNA, which will be used to carry amino acids, or it could be ribosomal RNA, 2 R's there, which is going to make up part of the ribosome. All right, so this is transcription. It occurs in the nucleus, and it's the process of copying DNA into essentially mRNA for our purposes. We now have a copy of the code, and we need to read that code, read the genetic code. And this diagram here just shows a simplified version showing how the DNA opens up or unzips at the location that it needs to, because that's where the gene is, and then RNA is produced. All right, so let's go on and have a look at translation, I imagine. Before we do that, just notice that this is the image that I tend to give out, and it's the one for the biology coloring book, and you just color this thing, and this shows transcription, RNA polymerase here in brown, and the ribonucleotides. Note that we got the base, we got the phosphate, but we have a ribose sugar. This is mostly linear. It doesn't stay attached. It doesn't stay in the nucleus. Goes off into the cytoplasm. And of course, you can make the transfer RNA and ribosomal RNA as well. Okay, here we go. Translation. All right. Now, I mean, I've been looking for other images of translation, and, there's lots of them on the internet, and they can be quite complicated. This is the one in the Nelson Biology book, so let's go through this one as best we can. This is called translation, and the idea is we need to translate a piece of mRNA. So this is our mRNA here, all right? And mRNA has these 3 base regions, which are called codons. Now, there is a small and a large subunit to a ribosome, all right? And during initiation, the mRNA will encounter the small subunit of a ribosome, and a transfer RNA carrying an amino acid, in this case methionine. This is the transfer RNA, okay? It looks like a boomerang here, and the amino acid's on the end there, and there are 2 sites on the large ribosomal subunit. There's the A site and the P site. During initiation, the first tRNA attaches to the P site, all right? And the anticodon here, in this case, I don't know if you can read that, but it says UAC. It is complementary to the codon AUG. So if the anticodon of the tRNA is complementary to the mRNA codon, then it will bind. And it happens to be carrying the correct amino acid. This is how the genetic code is read. If the codon and anticodon don't match, this is not the right amino acid to bring in. Okay, that was initiation. Now, in the next step, elongation, another tRNA comes in and it goes to occupy the A site. Think of the A site as like the additional site, okay, where you add in new tRNAs. So here we have tRNA1 and here is tRNA2 carrying the next amino acid. The anticodon CGU happens to be complementary to the mRNA codon GCA. Therefore, this is the right amino acid to be brought in. It happens to be alanine, all right? But you look this stuff up on the codon table. So we now have our 2 transfer RNAs and our 2 amino acids. We have to do that elongation step. And what we're going to do is we're going to transfer this amino acid over to alanine. That's going to take an enzyme. That's going to cleave this bond and then release this tRNA. It will have done its job. Now, let me analyze what has gone on here. In this diagram here, you can see that the methionine has attached to the alanine. There's a peptide bond here because these are amino acids. And this was transfer RNA 2 right here. The ribosome has moved down 1 codon, 3 bases, And a new tRNA, number 3, has come in and occupied the A site. It's carrying a new amino acid, and its anticodon is complementary to the mRNA codon. All right? These two amino acids, this dipeptide, will now be transferred. It always goes from the P to the A site, and a peptide bond will be formed. So that's what you see here, a tripeptide. And a fourth tRNA, this was 3, a fourth tRNA has come in because the ribosome moved down and that opened up that A site, and another tRNA has come in. The tripeptide will be transferred over to here. And this just continues and continues until you get to a stop codon, which does not call for any tRNA, and instead a release factor comes in and binds, which I guess they don't show here, and that signals the end of the polypeptide. Everything falls apart. Small subunit, large subunit with its sites, any extra tRNAs, and you now have your polypeptide chain. Or you have your final protein. This now, of course, remember, this has primary sequence of amino acids. Remember we studied that? This will then fold in various ways to give it its secondary structure, its tertiary structure. If it interacts with another chain, it'll have quaternary structure and all of that kind of stuff. This whole thing can be moderated. That would be translational control. Speeding it up, slowing it down, modifying it at the end, post-translational modification by adding functional groups here and, doing various things. Anyways, that's the process of translation. Pretty complicated and long-winded. Okay, let me see if I can take a break. Maybe I didn't record that before. All right, we're going to wrap this up anyhow. So I want to talk a little bit about the endocrine system and hormones. I want to also remind you that I've got other video tutorials on here. I've got synaptic transmission. I've got some other endocrine stuff under the endocrine histology lab. I've got some stuff on photosynthesis. So there's other videos that can help fill in some of the gaps for reviewing for exam, whatever. In terms of the major endocrine glands, we've got the male/female here. And we're going to start up in the brain region. We've got let's do the hypothalamus. Now, I know you can't see this that well, but the hypothalamus controls the pituitary gland that's inferior to it, all right? It has a number of releasing hormones that it's going to, if this is the hypothalamus here, and we got the pituitary gland here, releasing hormones are going to come down from the hypothalamus, that's H hypothalamus, this is pituitary. Releasing hormones are going to come down to stimulate the pituitary to release hormones, or inhibiting hormones will come down to tell the pituitary to stop releasing its hormones. And the pituitary, of course, has quite a few hormones, more hormones than any of these other glands. So that's how the hypothalamus controls the pituitary, although the pituitary itself is considered to be the master gland of the body. So the pituitary has all kinds of hormones. If we look just at one of them for the moment, let's look at TSH, thyroid stimulating hormone. This hormone comes from the anterior pituitary, and it comes down to the, whoops, sorry, I missed it. I was going for the thymus there. It goes down to the thyroid gland, and this hormone stimulates the thyroid gland to release thyroxine, or T3 or T4, and this is going to go off to most body cells and increase metabolism, metabolic rate, generate heat as a byproduct, use up oxygen. But this TSH, thyrotropin or thyroid stimulating hormone, is stimulated by TRH from the hypothalamus to be released. So you can see we've got 1, 2, at least 3 hormones in this long feedback loop. So that's one of them there. We also have ACTH, or corticotropin or adrenocorticotropic hormone, and this one comes again out of the anterior pituitary, and this will then target the adrenal glands, which sit on top of the kidneys here, and that will cause the release of glucocorticoid called cortisol. Glucocorticoid is a glucose regulating hormone from the cortex or outer region of the adrenal gland. ACTH is stimulated by CRH from the hypothalamus. So hypothalamus releases corticotropin releasing hormone. Corticotropin itself is then released from the anterior pituitary down to the adrenal, causing the release of cortisol, which is then going to go off again to body cells and cause glucose release. So there we see the hypothalamus again being involved. And I mean, we could just go on and on. Let's do GnRH. Gonadotropin releasing hormone from the hypothalamus goes down to the anterior pituitary, causing the anterior pituitary to release the gonadotropin hormones, or the gonadotropic hormones, follicle-stimulating hormone and luteinizing hormone. So that's a stimulation of those. Those will then affect the gonads, because these are called the gonadotropins. This is gonadotropin releasing hormone. These are gonadotropins. These will then go down to the gonads, either the ovaries or the testes, and cause sperm development, all right, sperm release, egg development, ovulation, egg release, that kind of thing. All right, so stimulation there. Of course, I'm not talking about the inhibitory hormones, which obviously do the reverse. So we've got those ones. We've got prolactin coming out of the anterior pituitary, PRL. There's, of course, a releasing hormone. So let's do PRLRH from the hypothalamus, stimulating the release of prolactin from the anterior pituitary, going down to mammary glands, stimulating them to produce milk, prolactation, the production of milk. There's a number of other hormones, growth hormone, etc., that are involved. If we have a look at a couple of the hormones that are actually produced in the hypothalamus but stored in the posterior lobe of the pituitary, if we look at the pituitary like this, looks like someone's foot, it's separated into two regions. That's the anterior part, that's the posterior part, right? Anterior faces the front, posterior sort of faces backwards. And the hypothalamus up here produces a couple of hormones which are transported down axons and are stored in axon endings right in the posterior pituitary. And we're talking about ADH, antidiuretic hormone, and oxytocin. So these are two hypothalamic hormones produced up here but stored in the posterior pituitary. ADH is released from the pituitary, from the posterior, and will travel through the bloodstream down to the kidneys and cause them to conserve water so that water is not lost in the urine. That raises blood pressure and keeps the body from being dehydrated. Oxytocin will come down to the uterus in the female and cause uterine contractions, and that again comes from the posterior lobe of the pituitary. We already talked about the thyroid gland, and on the back of the thyroid gland, you've got the parathyroid glands that release PTH, parathormone or parathyroid hormone. Those are involved with calcium metabolism and calcium regulation. PTH causes calcium to be increased in the blood, so it's going to come out of the bones. There's a bone, a very bad bone. So PTH will come down to the bone, causes calcium to be released out of the bone, and therefore it increases in the blood. And the other hormone that's antagonistic to PTH is calcitonin, which comes from the thyroid gland itself. All right, so I'll just go cal for calcitonin. That comes out of the thyroid gland. That goes to the bone as well, and that causes calcium to go into the bone, out of the blood. So this one will decrease calcium in the blood. This one will increase, the PTH will increase calcium in the blood. So they're opposite, they're antagonistic. Of course, we have a similar situation with the pancreas, where we have insulin, and insulin will target the liver and cause the liver, which is going to be in here somewhere, and it's going to cause the liver to absorb glucose, and that's going to decrease glucose in the blood. Whereas glucagon will also target the liver but cause it to release glucose. All right, so those are antagonistic hormones as well. And there are other antagonistic hormones and other hormones on here that I'm not talking about, because this video has gone on forever. You must be asleep. All right, thanks very much for watching. I hope that helps, and I wish you all the best for your exam. Take care.